The reason I decided to give this talk was because at the last Hope conference, I was in a bar with some guys after a long day of talks, and we were talking about the way information is encoded in various ways in various systems. And at the time, I was working on a DNA project trying to figure out how information was encoded in DNA. And I thought that they would be really interested in this idea, so we started talking about it. And there was clearly a lot of interest in it, but all throughout the conversation, they kept coming back to this, what's DNA? And I sort of had to realize that I was talking to people who, spent their lives setting up honeypots and filtering packets and stuff, and maybe they never took a biology course. The second reason I decided to give this talk is because I see a lot of similarities between what molecular biologists do in trying to figure out systems that are difficult to understand, and what other people do trying to explore systems. And I think that there's a sort of growing interest in molecular biology. And I think you can see that just in this next slide. Here's a cover of a recent issue of Make magazine in which there's an article talking about how to precipitate DNA in your kitchen. And this article describes how to isolate DNA, how to run a gel to characterize DNA, how to build a thermocycler to use PCR to amplify DNA. And what you see in the picture here is a glass with some ethanol on the bottom and some salt water on the top. And DNA is really soluble in salt water, but it's not very soluble in ethanol. So right at the interface, there's some precipitation going on, and you see these little tiny strands of DNA precipitating out of solution. So this leads to the question, what is biohacking? I think in general, it's two things for me. And one of them was, in honor of Stephen Levy, who spoke this afternoon. He talked about some of the characters in that book in the following way. They had grown up with a specific relationship to the world wherein things had meaning only if you found out how they worked. And how would you go about that if not by getting your hands on them? Now with biological systems, I tend to think they have meaning even if I don't really understand how they work. But if you consider that when you walk down the street, every leaf on every tree is executing operations that have been going on since longer than humans have existed on the planet, and we don't even understand most of those things. I think it's fascinating, and I think it's also fascinating to try to understand what those mechanisms are, and there are methods for getting your hands on the insides of living cells. And the second aspect of I guess, biohacking would be, given a little bit of information and given a little bit of knowledge, is there anything that I can do that's novel? I should say on that last slide, one of the main points I wanted to make here was that there was a time when the only people isolating DNA were PhDs and researchers, and now high school students are doing it. And I think these two groups of people are still isolating DNA, but they're doing it for two different reasons. Researchers are trying to find out more and more information about systems which they already know a lot about. Whereas a high school student might say, look, there's a lot of stuff I don't have to know to do something useful. And that's a form of abstraction. And abstraction is one way that engineers get things done. And this will come up later. So these two different aspects. One is sort of a top-down approach, trying to probe a black box and see what happens. And the other one is, if I have a few components, what can I do with this? I know what this protein is used for, but is there anything else I can do with this protein? So we could ask, are biological systems hackable? I think they are. Consider this picture here, where if you make a small change to the information content of a system, suddenly it has a very drastic effect. So this is a disease called brachydactylism. And a small change to the information content of DNA causes the second bone in your index finger to change length. So does that mean then that there's a gene which controls the length of every bone in your body? Is this information used anywhere else in your body? In other words, if you saw someone with a short index finger, would you expect their toes, their mama toe, also to be extra short? It turns out that people with this disease do have shortened toes, but you might ask, why doesn't it affect the length of the second bone of all of your fingers? It's a very similar bone, but it just doesn't. Another example of this is shown here, where a small change to the information content of a system causes an entire program to misfire. So on the left-hand side, on the left-hand side of this slide is a regular fly head, and there's these two little structures right here called antennae. But on the right-hand side, those antennae have been replaced by legs. So these two pictures are pictures of flies with legs growing out of their head instead of antennae. And it takes a developmental program to grow a leg. It's not just one, little change. So yet it's only one change in the DNA that causes this whole program to misfire. So I think these are both ways in which you can see that the information content of living systems executes very much like programs do, and things can go wrong and things can be altered. Both of these changes I just showed were things which were found in nature. Nobody knows enough about biology to go around giving people short fingers or make flies grow legs out of their face. But that's not to say that that's not changing. There's programs trying to figure out, like with salamanders, if you cut off one of their limbs, the limb grows back. In zebrafish, if you cut up some of their organs, the organs regenerate themselves. So these mechanisms exist, and there are research programs going on to try to figure out how do we, understand all this stuff. So in this talk, I'm basically gonna talk a little bit about DNA. I'm going to give a little bit of a historical perspective to try to set the stage for why is thinking about hacking molecules relevant now? What's changed in the past few years to make this such a pressing question now? And then I'm going to talk a little bit in detail about two biological circuits, just to sort of get a sense for what kind of things people do and maybe what they can do. So first of all, DNA is a molecule. It's in all your cells. Every living system is composed based on the DNA in their cells. All these organisms share DNA. In other words, they all have genes. The genes often will work between organisms. So these are yeast cells. This is a human being. There's a frog. This organism here is like a big nose with legs. It's got amazing olfactory capabilities. And the molecular mechanisms are often preserved between these organisms. So if you study the way cells divide in yeast, you can often understand mechanisms of cancer. So the molecular mechanisms are often preserved between cells, but the DNA content, these organisms exist because of the way that the information content in their DNA is executed. The way information is encoded in DNA is in the form of four what are called bases. And these are structures of those bases, A, G, C, and T, or A, C, G, and T. That stands for adenine, cytosine, guanine, and thymine. And these bases are linked together in chains to form a pattern. So they're attached to sugar molecules, and then there's a phosphate backbone which you can use to connect these things together. And once you can connect molecules like this together, you have a way of encoding information, because these things can stretch on for hundreds, thousands, and millions of units long. So it forms a way to make patterns. In cells, most of the time DNA doesn't exist as a single strand like that. Most of the time it exists as double strand. You've probably heard of the double helix. This is the two strands that are bound together, and they form this sort of twisty structure. But on that last slide where I showed one strand, because DNA exists in two strands, there's a set of pairing rules which determines how these two strands interact. So C's are always paired with G's, and A's are always paired with T's. The idea there is if you know the sequence of one strand, then you know the sequence of the other strand. The other thing to consider is that the strands are held together by a series of very weak interactions called hydrogen bonds. And these interactions are easily broken and can easily come back together. Whereas the chain that makes up the strand of DNA is formed by very strong bonds. So it's hard to break those chains. A fundamentally important property about DNA is its ability to come apart and then come back together. Because these hydrogen bonds are weak bonds, it's really no different than boiling water. If you add heat to water, the water turns into steam. If you add heat to DNA, it comes apart. But because there are these pairing rules, so DNA can melt and it can come back together, this provides a mechanism of addressability. So the pattern of bases on one strand will only come back together with its opposing pattern on the opposite strand. So this is the basis of a lot of things in biology. It's how organisms replicate themselves. It's how on CSI you can find some DNA at a crime scene and then go out in the population and match that DNA to some unknown population. So one of these strands, if put into a complex mixture of all of humanity, can find just the one strand that matches itself. This is also the basis of using DNA as a computer. If you could encode a problem in little snippets of DNA, you can generate the opposite strand as an infinite number of solutions. And if those little pieces can find the opposite strand in a library of infinite solutions, a certain structure will form, and you can isolate that structure, and this is how people use DNA as a computer. So it's a really powerful thing, but it's not very practical because it takes, with current methods, it takes about a week to solve a simple problem. If you write DNA out, like on paper, this is what it looks like. It's just a set, there's two strands, a top strand and a bottom strand. So it's not that readable, but if you are familiar with DNA at all, you can start seeing certain structures in here, like this ATG here, that's often the beginning of a protein. There's another sequence in here, GAA-TTC. This is a recognition sequence for an enzyme which can cut DNA into two pieces. So there's a lot of enzymes in biology which will recognize various signature sequences in DNA, so the DNA can be cut. And since it can be cut, there's also enzymes for putting it back together. And this is the way that you can sort of move genes between organisms, you can rearrange genes, you can make a lot of DNA structures using these enzymes. So I said that DNA encodes information, and the way that that information is expressed in the world is through something called the central dogma. So given a piece of DNA, if you want to express that DNA, the first step is to express it in the form of a single-stranded molecule called RNA. That's one level of gene expression, and it's tightly controlled by various things. The next thing is to then translate that RNA into protein. Proteins, they consist of 20 amino acids. The amino acid code is the same in all organisms. And proteins are the way that we get things done in the world. Basically, all these 20 amino acids have slightly different chemical properties. So it's the configuration of amino acids in a protein which gives you access to chemistry so that you can do stuff. All the DNA in an organism is something we call the genome. If you consider all the DNA that makes up a single organism, just represented as one long strand here. So here's a sort of a summary of the genomes of a few different organisms. E. coli is probably the most studied organism on the face of the earth. It's got about 4,300 genes, and its DNA content is about 4.5 million bases of DNA. Yeast and humans are both complicated structures that have nuclei. Yeast has about 6,000 genes. It's about 12 megabases. Humans are about 3 gigabases. But the number of genes is not really known because humans are complicated. And this number has ranged from 20,000 upwards of 150,000. And the best guess now is somewhere around 23,000. Yeah?
How about nematodes? Do you know offhand?
Yeah, nematodes have about 20,000 genes. So are we really no different than worms you find in your salad? I mean, this is the kind of thing you'd read in the paper, like, oh, how could we be the same as worms? But there's more to it than that, because it's not just genes. It's controlling how those genes are expressed. So consider that in 12 megabases, you've got 6,000 genes. In yeast, about half of that, so about half of the content of this DNA codes for those genes. So about 50% of the genome is codes for proteins, and about 50% is regulatory information or just spaces between genes. In humans, it's only about 3% of the genome which codes for genes. So 97% of the DNA is there, and we don't know what it's for. We don't know what it does. Why is there such a different density? And I've shown a little plot here. This is the yeast genome at 12 megabases relative to the human genome, just in length. Consider that DNA as a single molecule, if you were to stretch out all the DNA in one of your cells, it would be 6 feet long. And somehow all of your cells are able to wrap that DNA into a tiny little nucleus inside the cell. That's just fascinating. But given that the information content that organisms are determined by the information content in their genome, a few years ago people thought, well, why don't we just sequence genomes? At the time that genome sequencing was proposed, people were studying stuff one gene at a time. Yeah.
Questions. Do they refer to that 97% of the genome that doesn't specifically encode as junk DNA anymore, or is that?
Well, yeah. So the 97% of the genome that we don't know what it does, the term junk DNA is thrown around, but any serious scientist doesn't really, Oh yeah, it certainly does something because you can't do without it. So to call it junk is just like, well, you don't know what it does. I guess that means it's junk. I mean, it's not a very useful statement. What's that?